Restriction enzymes are of many types. One is restriction endonuclease, another is restriction exonuclease. So if you're talking about restriction endonuclease, I'm writing the spelling of it. Okay, restriction endonuclease is an enzyme which is very, very important for breaking the DNA at the restriction site. So the DNA is having many restriction site. The restriction site of the DNA is broken by restriction endonuclease. So restriction site, we told a number of times, it is a palindromic sequence. Palindromic sequence means, for example, A, T, T, G, G, C is there. Okay, C, right? This is the DNA. This is called what palindromic sequence. Again, I'll write for you A, T, T, G, G, C. Okay, T, A, C, C, G. So 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Right. So in this, which is looking exactly palindromic in nature, palindromic in nature. Any of you can say, okay, this is palindromic. 1 is not A, 2 is not C, 3 is not, okay, and this one, uh, T. Okay, then which one can be used as a palindromic sequence? This is not a palindromic sequence. So I am drawing, okay, why? Because many students are thinking the repetitive sequence. See, again, I'm repeating A, T, T, okay, T, T, A, right? G, G, C, C, okay, C, G, okay. So this is the palindromic sequence. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. So basically A always base pairs with thymine with double bond. G always base pairs with cytosine triple bond. So A always joins with thymine with double bond. G always joins with cytosine with a triple bond. So by keeping this, this is a palindromic sequence. So restriction enzyme will not break any DNA randomly. Restriction enzyme breaks a specific restriction site, which is having the mirror image of each other, which is nothing but palindromic sequence or inverted repetitive sequence. Inverted repetitive sequence. Again, I am repeating, inverted repetitive sequence is also called as palindromic sequence. Inverted repetitive sequence is also called as palindromic sequence. Is a part of the restriction site which is broken very easily by the restriction enzyme. Which is broken very easily by the restriction enzyme. Right? Now we are entering into the important concept. Restriction endonuclease and restriction exonuclease works in a different way. Okay, see, the restriction endonuclease is an enzyme which always tries to break restriction endonuclease enzyme, always try its level best to break the restriction site present in the center of the DNA. The restriction site present in the center of the DNA is broken by restriction endonuclease enzyme, right? So, whereas the restriction exonuclease enzyme breaks the restriction site present exactly at the end of the two standard DNA. So whatever the restriction site is present at the end of the DNA is broken by the again I am repeating the restriction site whatever it is present at the end of the DNA which is broken by restriction exonuclease enzyme. So restriction exonuclease enzyme is an enzyme which breaks the restriction site present at the end of the linear DNA. So restriction site present at the end of the linear DNA. So restriction site which is present at the end of the linear DNA is broken by exonuclease enzyme, whereas endonuclease enzyme is an enzyme which is very, very, very important to break the restriction site present at the inner point. So this is the way you're remembering different type of enzymes, restriction enzymes, right? And also I told about palindromic sequence n number of time. Again, back I'm writing the palindromic sequence A T T. So T A G. Okay. here G, G, C, 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 C. So this is the palindromic sequence. One, if it is there, here also one. If here above two is there, here two. If here three is there, the below also three. Like this, we can write all this palindromic sequence or inverted repetitive sequence. Again, like I'm repeating, restriction enzyme is an enzyme which breaks the restriction site present in the plasmid DNA, and plasmid DNA is nothing but cytoplasmic DNA. Plasmid DNA is nothing but a cytoplasmic DNA. So again, back I'm repeating, plasmid DNA is nothing but a cytoplasmic DNA to which we are introducing the plasmid DNA is also called as cytoplasmic DNA. Okay, by using this, we are introducing the gene of our interest into the host DNA. So host DNA is nothing but the target cell DNA. Host DNA is nothing but a target cell DNA. Into this host DNA, why I'm repeating 2-3 times? Why? Because for better incorporation of the message in your brain. So this plasmid DNA or cytoplasmic DNA is used to introduce the foreign gene into the host DNA. Into the host DNA. So this is possible with the help of the exonuclease enzyme and endonuclease enzyme. By using exonuclease enzyme, by using endonuclease enzyme, by using DNA ligase enzyme. So by using exonuclease enzyme, by using exonuclease enzyme, by using endonuclease enzyme, by using DNA ligase enzyme, I'm introducing gene of my interest with the host DNA, right? So now we are entering into the next important segment. DNA palindromic sequence. Now only I written for you a number of time for better understanding, right? If you're talking about here, this is palindromic in nature. This palindromic DNA is broken very easily by the restriction enzyme. Restriction enzyme. All restriction enzyme can't break all the restriction site. One restriction site, it belongs to type A. Other restriction site belongs to type B. Other restriction site belongs to C. Other restriction site belongs to D. A specific restriction site is broken by a specific restriction enzyme by using a specific methodology. By using a specific methodology. So all the enzymes will be not active in all the buffer system. All the enzymes are not active in all the pH, all the temperature. So all the restriction enzyme, by giving a proper pH and temperature, and we are giving the proper medium, some enzyme required mild acidic, some required strong acidic, some required less acid, less alkaline, some required maximum alkaline. So we are choosing the buffer with a specific pH. In that we are keeping the enzyme, restriction enzyme, to get the palindromic DNA sequence, to get the palindromic DNA sequence, which is having the credential, to undergo breakage. Okay, which is having the credential to undergo breakage. So can you see this DNA ligase enzyme is used for introducing the foreign gene between the broken DNA. So between the broken DNA or between the cleaved DNA, we are introducing the foreign gene by using restriction enzyme. By using restriction enzyme. That is a message we need to understand. Now, utilizing strictly the enzyme. The strictness of the enzyme with the complementary stick DNA facilitate the action of DNA ligase enzyme. We only told n number of time to you that if you are taking the double strand DNA, okay, to this double strand DNA, we are introducing the foreign gene. How we introduce the foreign gene? Okay, by using restriction enzyme and DNA ligase enzyme. So by using restriction enzyme and DNA ligase enzyme, by using restriction enzyme and DNA ligase enzyme. Again, I'm repeating, by using restriction enzyme. By using restriction enzyme and DNA ligase enzyme, what I'm doing is I'm introducing the gene of my interest into the plasmid DNA or vector DNA. This vector DNA introduces my gene of interest into the target DNA. To the target DNA, let us consider I'm introducing my target DNA or gene name called A. Okay, to the vector DNA or microorganism DNA. Can you give an example? All the microorganism I can't use. Only specific microorganism I can use. Bacteria, plasmid is a vector DNA. Bacteria, plasmid DNA is a vector DNA. It's a beautiful example. Okay, bacteriophage is nothing but a virus which infects the bacteria. So that bacteriophage DNA is vector DNA. Bacteria DNA is a vector DNA. Into this vector DNA, we are introducing the foreign gene of our interest, and that foreign gene of our interest is given back to the vector. Okay, again I'm repeating. The vector DNA or foreign DNA is having the DNA of my interest that is used to get incorporated into the target DNA. Now the target DNA is having the gene of interest. So now we are entering into the next important segment, gel electrophoresis. Before entering into the gel electrophoresis, in the lower grade we studied the gel electrophoresis is a technique used to separate the particle of various size and shape. So particle of various size and shape. So particle, again back I'm repeating, particle of various size and shape. So particle of various size and shape we are separating by using the electrophoresis. We are separating by using gel electrophoresis. So for example, one DNA fragment is having the length of 12 base pair, okay, and another DNA which is having the length of 7 base pair, okay, compared to 700 base pair, 12 base pair. So we're having 7 base pair, right? So the DNA having 7 base pair length will move very fast. The DNA which is having 12 base pair length will move very slowly. Will move very slowly. Why? Because the gel is having a lot of porosity in it. The gel is having a lot of porosity in it. So through the porosity of the gel, the DNA length of various size and shape, we can make it to get migrated and we can make it to get separated, right? In the gel medium. Gel is transparent. So in this transparent gel, the moving DNA is visualized. Why? Because very easily, okay, before adding the DNA into the gel medium, we are using important chemical called yttrium bromide. So yttrium bromide is a chemical which is used uh, for staining the DNA. So yttrium bromide DNA is used to run in the gel. That means first thing is, for example, a specific drumstick plant DNA. I am breaking into small pieces by using restriction enzyme, and then I'm adding yttrium bromide to it, and then the colored yttrium, the colored DNA or yttrium bromide colored DNA you say, or yttrium bromide colored DNA you say, and that particular DNA fragment you run in the gel that we can visualize it. So any one of you or having any doubt in the restriction enzyme, please let me know. We know the visualization of the DNA fragment is possible. Visualization of the DNA fragment is possible. Okay, in the gel. So the strained or colored DNA is moving the gel. The strained or colored DNA is moving the gel. Okay, that we can visualize by because we already colored the broken DNA by using the restriction enzyme. By using the restriction enzyme. By using the restriction enzyme, we already colored the DNA, right? And that color DNA is made, or else we can say the color DNA we are running the gel. After running the gel, the color DNA intensity we are understanding by passing the UV light. So the color DNA with yttrium bromide, once you are coloring it or staining it, the color DNA after running the gel, we are the color DNA after running the gel, we are passing the UV light over it, and then we are understanding the DNA intensity. If the DNA intensity is very high, if the DNA intensity in terms of absorption and reflection, then the DNA is very, very long. Then the DNA is very, very long. So some DNA is having the short length, some DNA is having the long length. So again, I'm repeating, one is called what? Short DNA, another is called what? Long DNA. One is called short DNA, another is called long DNA, right? So to the short DNA and long DNA, to our eyes it is not visible. So we are using a dye called etherium bromide. This etherium bromide chemical is very, very important for coloring the DNA. Etherium bromide is used for coloring the DNA. So etherium bromide is used for coloring the DNA. After coloring the DNA, we are making the DNA to move in the gel, and then over the color DNA, we are passing the UV light, and we are getting the uh, reflected intensity, right? So the reflected intensity or the light is measured, and then by which we are understanding the DNA fragment length. So then we are removing the dye. That is called elution. Before entering into the cloning vector, okay, you know very well the related videos are there. The related videos, the tools for the recombinant DNA technology, you can see very nicely explained. And they're using one particular microbe DNA, one particular microbe DNA. So this is the one particular microbe DNA into which we are introducing the foreign gene. Okay, the foreign gene is this. For example, this gene is an insulin gene. So this insulin gene I introduced into the microbial DNA, right? By using, okay, by using which particular enzyme? By using restriction enzyme. Again, back I'm repeating, restriction enzyme is an enzyme which you are using to break the DNA. After breaking the DNA, we are introducing the foreign gene of our interest by using DNA ligase enzyme. So by using DNA ligase enzyme and by using restriction enzyme, I introduce the insulin gene into the microbial DNA. What microbial DNA? Can I say any one specific word? Bacteria. So in the case of bacteria, what DNA you are choosing? Cytoplasmic DNA. How do you call the cytoplasmic DNA as a plasmid DNA? So to the bacterial cytoplasmic DNA or plasmid DNA, to the bacterial cytoplasmic DNA, to the bacterial cytoplasmic DNA or plasmid DNA, we introduce the microbial DNA by using restriction enzyme and DNA ligase. And now this bacterial DNA, uh, here is this here, and to this DNA we are introducing the insulin gene. And then this is a host DNA. This is a host DNA. To the host DNA, what is exactly happening here is this is a host DNA. Into the host DNA, the insulin gene is introduced. To the host DNA, insulin gene is introduced. Again, back I'm repeating. To the host DNA, insulin gene is introduced by using this bacteria. So the bacteria introduce the insulin gene of my interest to the host DNA. Okay, and now the host DNA is an insulin gene. This is one way of cloning process. Second way of cloning process is this entire bacterial DNA with the microbial DNA I am introducing into the bacteria. If the bacteria multiplies, plasmid also multiplies. If the plasmid multiplies, insulin gene also multiplies. After the insulin DNA multiplication, what is exactly happening? We are getting the insulin RNA. That insulin RNA is making the insulin peptide, which is ready for harvest. In the sense, we can isolate from the microbe for usage, specifically to treat the diabetic patient, right? So why I need to do this? Once upon a time, they use a bear to produce insulin. They'll kill the bear. They'll take the pancreas from the bear, and they will take the extract of the pancreas. That extract is used. Okay, how many diabetic patients are coming to the world in each and every day? You know this. Per actually, the World Health Organization is saying every 100 of the world is having one diabetic patient. Every 100 of the world population is having one diabetic patient. So to satisfy those many number of diabetic patients, I can't kill that much bear. That many number of bears are not there in the world. So what we are doing, we are using bacteria to make the insulin, right? And now what we need to understand. So basically, one human is coming to the world with dwarfism. I can't grow furthermore. You not to worry about it. We are here with you with the biotechnology tools, and then we are ready to make the growth hormone. at the massive level at a very cheap price for you, so that what we can do is this hormone we can give for you for your better growth, so the dwarfism will be reduced. Okay, okay, that is our goal, right? So how we made the growth hormone? The growth hormone gene we introduced into the bacterial DNA, and we made the bacteria to grow with this growth hormone gene. And that growth hormone is not required for the microbe. This growth hormone is released by the bacteria. Bacteria release the growth hormone. Who release the growth hormone? Bacteria release the growth hormone. Bacteria release the growth hormone. Again, back I'm repeating. Bacteria synthesized and release the growth hormone. Bacteria synthesized and release the growth hormone, right? And this growth hormone is produced with the bacteria because we incorporated the growth hormone gene into the bacterial DNA. So we introduced the growth hormone gene into the bacterial DNA, and that bacterial DNA made the growth hormone, which is not required for the bacteria. So it releases outside of its body. So that is a major resource for us for this hormone, which we are purifying it and we are selling the vials after appropriate preservation and uh, at a proper temperature control. That is very, very important, right? So now we will be entering into the tools for the biotechnology. In this, the first thing is we are seeing the cloning vectors. Basically, the vectors, the word we are using in the case of microbiology, n number of time to specify, you know very well, the microbe. If you're talking about the microbe, uh, with disease causing efficiency. Okay, for example, my name is female Anopheles mosquito. I'll transmit malaria causing protozoa. So malaria causing protozoa, I'll transmit. So the malaria causing protozoans. Okay, I'll transmit. Who am I? My name is protozoa. Okay. So again, back repeating, plasmodium. Plasmodium is a type of protozoa that is transmitted by Anopheles mosquito. So female Anopheles mosquito transmits plasmodium. So in this regard, I am calling this mosquito as vector, but that is not the meaning here. In the cloning vector, max vector is different. Physics vector is different. Okay, microorganism vector for disease is different. In biotechnology, vector is different. All vectors are not same. All vectors are not same. Here, what is vector? Can you tell me in detail? We are using microbial DNA for introducing gene of our interest into the host DNA. So always we'll use bacterial DNA? No. Sometime I'll use phage DNA. Sometime I'll use fungus DNA. Sometime I'll use bacterial DNA. It depends upon my need. Okay, sometime I'll use bacteria. So phage is not a bacteria. Phage is nothing but a virus, right? Sometime I'll use phage DNA. Sometime I'll use fungus DNA. Sometime I'll use bacterial DNA to introduce the gene of interest into the target DNA. Into the target DNA. Again, back I'm repeating, to the target DNA, I'll introduce gene of my interest. To the target DNA, I'll introduce the gene of my interest with the help of the fungus or bacteria or virus. So, in this regard, I am calling microbial DNA as a cloning vector. I'm calling microbial DNA as a cloning vector. So, the crux of all this sentence is given properly with multiple examples for better understanding by our Gotouniversity team. Now we need to understand basically the alien DNA, how we are duplicating. We can duplicate the alien DNA of our interest. For example, I need to duplicate the insulin gene. I need to duplicate the thyroxine is nothing but a thyroid hormone making gene. So thyroxine is a hormone. This thyroxine hormone I can make to the maximum level, right? So here, depending upon my need. So insulin hormone or thyroxine hormone. Sometime I need to make growth hormone at the massive level. So what we need to understand basically growth hormone I need to make at the massive level. So growth hormone production I can increase. Thyroxine hormone production I can increase. Insulin hormone production I can increase. So by using the microbial DNA as a vector, by using microbial DNA as a vector, I can increase the hormone production. I can increase hormone production. And second thing is, if you're talking about this gene, this microbial DNA is having this insulin gene. Why? Because we introduced it. Sometimes we'll do mistake. So insulin gene we are keeping as multiple copy. So thyroxine gene we are keeping as a multiple copy. How this is possible? By using PCR technique. So either you use PCR technique or either you use microbial DNA to duplicate the given gene into multiple copies. To duplicate the given gene into multiple copies. To duplicate the given gene into multiple copies. Again, back I'm repeating. To duplicate the given gene into multiple copies, either you use microbial DNA or either you use PCR. Either you use PCR or either you use microbial DNA, okay, to duplicate the given gene into multiple copies. Why? Because while doing the experiment, we'll do mistakes. So the gene will go for breakage. The gene will go for breakage, right? So this is the way you need to remember. And now, calmly, you listen to me. In the microbial DNA, this insulin gene or thyroxine gene or growth hormone gene, we are multiplying. In PCR technique, also we can duplicate the given gene into multiple copies. So after duplicating the given gene into multiple copies, we are keeping those genes as a DNA bank. Okay, we are keeping the particular character controlling gene as a DNA bank. So DNA bank, a DNA bank for a character, we are maintaining. DNA bank for a character, we are maintaining. So our DNA bank for a trait, we are maintaining. For what for we are maintaining? Well, because we need to um, ensure the production of the insulin. We should produce thyroxine hormone. For that, we are maintaining the DNA bank for insulin. We are maintaining the DNA bank for thyroxine. So by duplicating that given gene into multiple copies by microbial DNA and by PCR technique, right? So here, see in this place, by seeing bacteriophage, what is coming to your mind? So the bacteriophage is nothing but a virus which infects the bacteria, which is not given in the slide. So virus which infects the bacteria. There are some students or they're in this class, okay, just they are taking a snapshot of the notes, okay. But we design in such a way, being a Gotouniversity team, so we need to bring a lot of sentence, hidden sentence for each and every sentence. All the points are not here. All the points are not given here. Okay. Bacteriophage is nothing but a virus which infects the bacteria. What do you mean about plasmid? Cereblasmic DNA. What do you mean about cereblasmic DNA? Double stranded DNA. Where it is present? In the bacteria. We can use this plasmid in virus? No. We can use the plasmid word in the case of yeast? No. Where I can use? Only for bacteria. Only for bacteria, I can use this plasmid DNA. Is the DNA of the bacteria. It's the DNA of the bacteria. So for each and every word, hidden sentence are there. Plasmid means DNA of the bacteria, and that too cereblasmic DNA. Bacteria, cereblasmic DNA is plasmid DNA. Bacteriophage DNA is nothing but the DNA of the virus which infects the bacteria. Okay, so all the sentence will not give all the information in one stitch in one go, right? And we need to understand basically the bacteria DNA and that too cereblasmic DNA is having one credential. So whereas virus DNA is having other credential. What credential we'll discuss? See, we know very well the phage DNA is having one credential. What credential? It can duplicate the big size gene. The phage DNA can duplicate the big size gene. Phage DNA can duplicate the big size gene. Phage DNA, again back I'm saying, phage DNA can duplicate the big size gene. Plasmid DNA will duplicate the small size gene. Small size gene. So these are the hidden sentence which is not given here, right? So whenever you are talking, we are talking randomly. No, in this year this tough question came. In that year that tough question came. So that question we are running in our mind, and then we are talking. Okay, here plasmid and bacteriophage. possess the ability to replicate independently. That is already given here in the slide. That is already given in the slide. For reading the sentence we are here, no, no. Small size gene duplication is possible with the plasmid, but not big size gene. Bacteriophage can duplicate the big size gene, but not at a fast rate. But not at a fast rate. I'm a phage DNA. I'm a bacteriophage DNA. I can duplicate the big size gene into many copies, but at a slow rate. Plasmid DNA, which duplicates a small size gene at a fast rate. So the duplication of the small size gene at the fast rate is possible with the plasmid. The duplication of the big size gene at the slow rate is possible with the phage DNA. That is the reason why this plasmid bacterial DNA and phage DNA are integrated by the great scientists of biotechnology industry. So that we used to call as cosmid as a DNA, which is very, very important to duplicate the gene of any size into multiple copy at a fast rate. So this is called cosmid DNA. So this cosmid DNA is a hybrid DNA, which is not a phage DNA, which is not a plasmid DNA. It is hybrid DNA of both the species, right? So these are the hidden sentences for each and every sentence. We're not given everything here. We're not given everything here, right? So now we need to understand the plasmid can vary in the copy number. So 15 to 20 copies are formed in multiple number and even more per bacterial cell, right? So these are the crux, all this crux essence we already narrated in the whiteboard area. So now, if you're talking about the phage DNA, as I already told you what credential it is having. This bacteriophage is a virus that infects the bacteria that is very, very similar to tobacco mosaic virus in terms of morphology. In terms of morphology, tobacco mosaic virus, how it looks. In that way, the bacteriophage is not okay. Most of the bacteriophage looks in this way, right? All the virus are not having the same size and shape. Or not having the same size and shape. If you're seeing the bacteriophage, immediately the image of the bacteriophage should come to your mind, right? So the image of the bacteriophage should come to your mind, right? So this bacteriophage is having this is the head segment and this is the DNA segment of the phage. So bacteriophage means sometimes what will happen is they will give the bacteriophage picture to you. They will not use bacteriophage. You need to say, what is this? This is what a bacteriophage, a virus which infects the bacteria, right? And then sometime what they will do is, can you see this by seeing this picture? So listen very, very carefully. By seeing this picture, what they will do, I'm zooming it. Can you see this? They will not give anything to you. They will not give anything to you. So this is a phage. Can you see this? This is a phage DNA. So this is a bacteria having flagella, having flagella. Can you see this? This DNA, this DNA is a bacterial genome. Bacterial genome. Cytoplasmic DNA or nuclear DNA, they're not mentioned now. See, can you see this? In the bacteria, they told bacterial genome. They told bacterial genome, that's it. Okay, okay, how many different type of bacterial genomes are there? How many different type of bacterial genomes are there? Like this, if they're saying how you will say all this information. Um, how many different type of bacterial DNA? See, one is called nuclear DNA. One is called nuclear DNA. Another is called cytoplasmic DNA. See, listen, I'm feeling that tomorrow morning I'm having the board exam. Tomorrow morning I'm having the board exam. If they're asking the two different DNA or the bacterial genome, what I will write in the board exam? That is not there in the slide. Listen to my speech. Write it down in the note. Okay. The nuclear DNA is a DNA which is nothing but the linear DNA. Cytoplasmic DNA is a circular DNA. There is a lot of difference between linear and circular. And again back, this cytoplasmic DNA is double standard. Okay. And this nuclear DNA is also double standard. Linear, double standard nuclear DNA. Okay. Circular, double standard cytoplasmic DNA. Circular, double standard cytoplasmic DNA. Circular, double standard cytoplasmic DNA. Again back, I'm repeating. circular double-stranded cytoplasmic DNA, linear double-stranded nuclear DNA, linear double-stranded nuclear DNA, right? Linear double-stranded nuclear DNA, linear double-stranded nuclear DNA. Okay, cytoplasmic circular double-stranded DNA, circular cytoplasmic double-stranded DNA. Okay, linear nuclear double-stranded DNA. So these two different type of DNAs are there. So they will ask in the board exam, okay, can you differentiate or can you narrate or can you say the types of the bacterial genome? You need to say this too. You need to say this too. So the class, okay, here in the case of online class, okay, though the biology teacher is sitting in the comfort zone, sitting in the cushion seat and taking the classes, okay, it's not like this. Okay, we need to study a lot of board papers, right? And we are discussing all the hot questions of not only the NCRT example, NCRT exercise, and also given by the CBSE board, right? Okay. Now we need to understand the next important concept. Again, I told a lot of pros and cons of all these things in detail. Okay. Now, calmly you listen to me. What is meant by recombinant? What do you mean about recombinant? Any one of you can say this? Okay. You know very well the recombinant is having the alien DNA. They already had given having foreign gene. Sometimes they will say the DNA which is having one more other DNA which is not the part of the same species. Alien DNA, so species one DNA. So how you will say different recombinant? Okay, different recombinant if they are saying so own DNA of the species is having foreign DNA as well for bringing the reservable character. Like this, we need to keep the dot if it is 1 mark. If it is 2 mark, you need to write the example. If it is 3 mark, you need to write the explanation for the example. Okay, explanation for the example, right? So again, like I'm saying to you, one is answer, another is after the answer, we are giving the example. After the example, we are giving explanation. If it is 3 mark, if it is 3 mark, we need to give like this. Sir, can you say the answer? Different recombinant? Recombinant DNA is have the alien gene in it. Example, example, bacterial plasmid DNA. Example, bacterial plasmid DNA having insulin gene. 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 Okay. By example, bacterial plasmid DNA having insulin gene. Explanation, insulin gene or the bacterial plasmid DNA which makes the insulin hormone which is not required for the bacteria. So bacteria releases it outside in the bacterial culture medium wherever it is growing. Bacterial culture medium wherever it is growing. The bacterial culture medium wherever the bacteria is growing, there it releases the insulin hormone. So now we are coming for the next major segment is bacteriophage has unique ability to replicate. Bacteriophage is having the unique ability to replicate independently with the bacterial cell. Independently with the bacterial cell. So, um, so who is having the independent replicating property? Bacteriophage is having the replicating property. The bacteriophage is a virus which can duplicate inside the bacteria into multiple copies after bringing the bacteria genome in its control. The phage DNA is controlling. Phage DNA is controlling the bacterial genome. Phage DNA controls the bacterial genome. Phage DNA controls the bacterial genome, and then the bacterial genome. Again, I'm repeating. Phage DNA controls the bacterial genome, and then compels the bacterial genome to make the phage protein. So phage protein, phage RNA, phage DNA, phage protein, RNA, DNA, everything are formed. So phage DNA, phage RNA, phage protein. So phage DNA, RNA, protein, all these things are formed, which are combining and making the phage. So who made this bacterial DNA? Who corrupted the bacterial DNA? Phage DNA. Phage DNA corrupted the bacterial DNA. Bacterial DNA corrupted the phage DNA. No. Phage DNA corrupted the bacterial DNA or bacterial genome. As a result, phage DNA, RNA, protein are formed and many phage are developed. Many phage are developed. So the phage structure we already drawn for you a number of time for making you to understand about the phage morphology. Phage morphology, right? So now what we need to understand basically the genetic engineering of the bacteria, right? Enables the multiplication of the alien DNA. Alien DNA. So we need to understand basically the plasmid or bacteriophage DNA with a vector designed to facilitate the process and to select the recombinant more effectively. Recombinant more effectively. So uh, basically, phage DNA is very important. Again, by repeating, phage DNA is very important in incorporating the foreign gene of interest into the host DNA. Into the host DNA. Right. And second thing is here in this particular contest, who is the host? Who is the host? Who is the host? Who is the host? Host is bacterial DNA. Host is bacterial DNA into which who is incorporated, into which component has introduced, the foreign gene has introduced. The foreign gene, let us consider the insulin gene. Who has introduced it? Phage DNA has introduced it. Phage has introduced to the host DNA. In the host bacterial DNA, okay, the insulin is introduced by the phage DNA. Okay, phage DNA, right? So they will ask how we can introduce the insulin gene into the bacteria, how we can introduce the foreign gene into the bacteria without the plasmid destruction, without the plasmid destruction checkpoints they will think. So without the removal of the plasmid from the bacteria, how we can introduce the gene of your interest? Like this, if they're asking, it is too difficult for us to think and write. That answer only be given here. The phage DNA, we having the insulin gene, is having the potential to incorporate the insulin gene into the host DNA. To the phage DNA, we introduce the insulin gene by using two important tools. One is called restriction enzyme, another is called DNA ligase enzyme. By using restriction enzyme and DNA ligase enzyme, by using restriction enzyme, by using restriction enzyme and by using DNA ligase enzyme, by using restriction enzyme and DNA ligase enzyme, we are introducing insulin gene into the host DNA. We are introducing the insulin gene into the host DNA, right? We are introducing the insulin gene into the host DNA. Now, again, back I'm repeating, we are introducing the insulin gene into the host DNA, right? Now we are entering into the next major concept is the cloning vector plays a crucial role in the genetic engineering. Insertion and replication of the foreign gene into the host cell, right? So how we can introduce the foreign gene into the host cell? See, for example, let us consider here I am a very good Carnatic singer, right? So there are some famous piano specialist, violin specialist, okay, guitar specialist, mridangam specialist, tabela specialist. Everybody are there from different countries. So we are all assembling here, and then we are all doing so many number of programs, rehearsal, and then we are fixing all of our mind alignment in the same lane for one particular song. In the same way, if I need to introduce the foreign gene into the DNA, I need to be very careful by satisfying by satisfying all the members. Satisfying all the members. For example, let us consider, okay, in your DNA, if I need to introduce um, the in a DNA, if you need to introduce uh, the foreign gene. So in the plasmid, if I need to introduce a foreign gene, I need to consider origin of replication. That is nothing but ori.c. Okay, in some book they will write ori. In some book they used to write ori.c in the international books. IB curriculum, you can see ori.c. Instead of saying ori.c, you can say replicon, also called as origin of replication. Origin of replication is also called as replicon, or ori.c is also called as replication initiation point. So replication initiation point or ori.c is also called as replicon. See, these are all the things why we need to say. Why we need to say multiple terms they are using the board exam, which is not there in the slide. ori.c is there? No. Replicon is there in the slide? No. Replication initiation point is there in the slide? No. We need to come with preparation to the class, and you need to revise before coming to the next class whatever I taught today. Like this, have a routine academic cycle planner. In this origin of replication, where the foreign DNA is linked to the sequence. When the foreign DNA is linked to the sequence, what happens here is inside the host cell, the foreign DNA is getting duplicated. The foreign DNA is getting duplicated. For example, this is the foreign gene. I am keeping as F gene like this. I am keeping this as host DNA. So I am keeping this host DNA into which we introduce a foreign gene by using two molecular enzyme, which is not only restriction enzyme, which is also DNA ligase. By using these two enzyme, I introduce a foreign gene into the host DNA. Okay. But whenever I'm introducing here, the origin of replication should not get inactivated. The origin of replication should not get inactivated. For example, when I'm giving the vocal vibration, when I'm singing one song, okay, here my tabla specialist is saying this tone is not suiting my beat. Okay, can you change the voice? Yes, of course, I'll do it. Like this, I need to address them. The guitar specialist is saying, okay, a little bit, can you increase the speed of your pronunciation? Like this, if they're saying, so I need to increase the speed of my pronunciation. So all the specialists, the music specialists, how they are helping me to bring a great song, the same way in a plasmid DNA, when I'm introducing the foreign gene, the origin of replication should support this introduced foreign gene. The promoter should support this foreign gene. Okay, again back I'm repeating, promoter should support the foreign gene, origin of replication should support this foreign gene. Okay, and then the second thing is regulator should support this foreign gene. Again back I'm repeating, regulator. So regulator should support the foreign gene introduction and multiplication. Promoter gene should support the foreign gene introduction. Origin of replication should support the foreign gene introduction and multiplication. Host DNA should support. So by keeping all the associated genes to the plasmid, we are introducing. So origin of replication is very, very important. Why it is important? Why it is important? Okay, why? Because the origin of replication after getting disassembly, after facilitating the disassembly. For example, the double-stranded DNA facilitates the origin of replication to undergo unwinding. From the origin of replication, unwinding happens. Unwinded DNA. Again, back I'm repeating, unwinded DNA. Unwinded DNA makes the new DNA synthesis. Unwinded DNA makes the new DNA synthesis. So this unwinding is initiated only from the origin of replication. So who need to create the origin of replication during the DNA synthesis means the DNA polymerase enzyme. So we need to understand DNA polymerase enzyme is an enzyme which creates the origin of replication. Like this, we should not say DNA helicase enzyme. So DNA helicase enzyme is very, very important for creation of the origin of replication before the DNA synthesis process by DNA polymerase. By DNA polymerase. I'm a DNA polymerase. I need to duplicate the given DNA segments into multiple copy. For that, the unwinding should happen. How the unwinding happens? Because of the DNA helicase, it happens. Okay. So this DNA helicase causes unwinding in a spot called origin of replication. So what will happen is, sometime if you are introducing the foreign gene, the origin of replication get collapsed, regulator get collapsed, promoter get collapsed. So we should be very, very careful when you are doing it. Now we are coming for the selectable markers. See, for example, I'll say the selectable markers application in disease, then I'll come to biotechnology. For example, one particular woman is having the breast tumor. In that breast tumor patient, the unusual protein BRAC21 is produced by the breast cancer patient. So BRAC21 protein is produced by the breast cancer patient. That is a indicator for the cancer understanding in terms of progression level. So the tumor has grown this much level. So accordingly, I need to give the treatment. For that, I am doing the quantitative analysis of the BRAC2 protein in that particular breast cancer patient. So like this, here also selectable markers are there. For example, in a E. coli DNA, whatever the foreign gene we introduced, which is existing in the E. coli DNA or not, I don't know. So for that, there is some cross-examination screening procedure is there. So again, back I'm repeating, this is the E. coli DNA. This is the E. coli DNA. Into the E. coli DNA, we introduce a foreign gene of our interest, which is nothing but the insulin gene, which is nothing but the insulin gene. So what we need to understand is, this is nothing but insulin gene. This is nothing but the E. coli DNA. So what happens exactly? This, let us consider the E. coli DNA is having one more gene. The E. coli DNA is having one more gene. So if we're talking about this gene name called the gene of the E. coli, which is making one enzyme called beta galactosidase enzyme. Beta galactosidase enzyme. This beta galactosidase enzyme is an enzyme which is responsible for breaking the lactose, for breaking the lactose, for breaking the lactose. So again, I'm repeating. So here, calmly listen to me. Okay. So E. coli DNA is this. The E. coli DNA is having the foreign gene or not, I don't know exactly. So to cross-examine it, okay, what I'm doing one by one, I'll say to you, listen very, very carefully. The insulin gene is incorporated between the gene which is responsible for making beta galactosidase enzyme. Beta galactosidase enzyme is an enzyme which is very, very important for the E. coli to survive by breaking the lactose into glucose and galactose. Okay, lactose is broken into glucose and galactose. This is because of the beta galactosidase enzyme. So this enzyme is not produced. This enzyme is not produced. This enzyme is not produced. This enzyme is not produced if we are introducing insulin gene. So if the lactose is not getting converted into glucose and galactose, this enzyme is not working properly. Why the beta galactosidase is not working properly? Why? Because the insulin gene is incorporated. Okay. Okay, to which particular DNA? To the bacterial DNA. So if I am not getting glucose and galactose in the bacteria culture medium or bacteria growth medium, so that means my bacteria DNA is an insulin gene. Bacteria DNA can't make glucose and galactose. Lactose is not broken. Why? Because insulin gene is there. So this is the way I am selecting the uh, recombinant. Means the recombinant having the some other gene which is not relevant to that. So the bacterial DNA don't want insulin gene. That insulin gene, but required for us, that is there in the bacterial DNA. So that I can understand by not getting galactose. By not getting galactose. So this is the way we are using selectable markers, right? And then other information is also there. If you are talking about the calm level of understanding is very, very important, right? So this is the insulin gene. Okay. See, this is the bacterial DNA. This is bacterial DNA, right? So in the bacterial DNA, this bacterial DNA, this is the antibiotic breaking enzyme. Antibiotic, antibiotic breaking enzyme. So antibiotic breaking enzyme gene is this. That gene name I am calling as AA. So this is the bacterial DNA, okay, into which I introduce the insulin gene. I introduce the insulin gene. So the insulin gene is this. The insulin gene is this. So this insulin gene, which is introduced within the antibiotic breaking enzyme making genes. Antibiotic breaking enzyme making gene into which I introduce the insulin gene. So here bacteria is not breaking antibiotic. In the culture medium, I kept a lot of antibiotic. The antibiotic is not broken. So bacteria is not growing properly in one place. That means that bacteria is having my insulin gene. Again, back I'm repeating. Okay, in some place, the bacteria is growing very nicely. Why? Because that bacteria is having antibiotic breaking enzyme much more. Whereas in other places, for example, here the bacteria is growing much more. Okay, here the growth is very, very high. Here the growth is very, very less. Why the bacteria growth is very, very less here in this place? Why the bacteria growth is very, very less in this place? Why bacteria growth is very, very less in this place? Why? Because this bacteria, again back I'm repeating, why the bacteria growth is very, very less in this place? Why? Because that is struggling a lot in the antibiotic medium. Why it is struggling a lot? Why? Because my introduced insulin gene is disturbing this gene. That means I need this bacteria. So I need this bacteria. So this bacteria can't make enzyme to break antibiotic. That means it is an insulin gene. This bacteria I'll grow into millions and trillions. And then that bacteria produces insulin hormone. That is a way we are selecting the bacteria, right? So be very careful with all those things. Now we are going for the cloning sites. If you're seeing the cloning site, okay, this is a plasmid DNA. This plasmid DNA, why we are coining the term PBR 322? Because Bolivar and Rod Rigornus are the two great scientists. Can you see this place here? Anywhere they given the expansion of PBR 322? No. Anywhere. Okay, PBR indicates Bolivar and Rod Rigornus. Is it indicated here, written here? No. So, what is the purpose of saying that we are great? We are not saying. The thing is basically, um, the concept is applicable for you as well as for me. Okay, for example, if you're pointing HIN3, what is the expansion of HIN3? Is not there in the slide. E restriction enzyme site for Haemophilus influenza. Restriction enzyme of Haemophilus influenza is breaking the site. Restriction enzyme of Bacillus aminoleucophacium is breaking the site. This is ampicillin resistant gene. This is tetracycline resistant gene. This is origin of replication. Restriction enzyme of Escherichia coli. Restriction enzyme of Clostridium. So like this, what I'm saying everything, right? So be very, very careful. So by practice only it will come. Sitting alone and with a lot of motivation, with a lot of what active mindset. If you're studying, then the content will remain in your brain as evergreen material, right? So how to operate your brain is also very important. So whenever I am teaching Haemophilus influenza. Like this, I'm teaching. No, you know very well Haemophilus influenza is a bacteria having the DNA, having the DNA breaking capacity. Haemophilus influenza is having a restriction enzyme. Haemophilus influenza is having the restriction enzyme. The restriction enzyme of Haemophilus influenza, okay, is called what? HIN2. This site is broken by restriction enzyme of Haemophilus influenza. So this site is broken by restriction enzyme of Bacillus amyloleucophacium, right? So what I'm saying, the thing is, by Okay, in the board exam, they will confuse us with many way. Okay, by showing the pictures in detail. By showing the picture in detail, so by showing the pictures in details, right? Um, here, this is the plasmid DNA. In the plasmid DNA, I told about the origin of replication. Okay, the place where the DNA is getting unwinded. Yes, of course, the DNA should get unwinded. Then only by using the unwinded DNA, new DNA is formed. Then only by using the unwinded DNA, new DNA is formed. Right? By using the unwinded DNA, which DNA is formed? The new DNA is formed. So, origin of replication is very, very important. Right? So, each and every site denotes the restriction site. That restriction site is broken by a specific restriction enzyme of a specific microbe. So specific restriction enzyme of the specific microbe, specific restriction site is broken by specific restriction enzyme of a specific microbe, right? Okay. So specific microbe having specific restriction enzyme to break a specific restriction site. So each one of the box, whatever I drawn here, or the circle is nothing but restriction site broken by the specific restriction enzyme of a microbe. That is a message you need to understand. Now, this PBR 322 is a plasmid of the bacteria designed by Bolivar and Rodriguez. So this ampicillin and then tetracycline resistant genes are present. So ampicillin and tetracycline resistant gene. So ampicillin resistant gene. Ampicillin resistant gene. Again, back I'm repeating, ampicillin resistant gene is a gene. Ampicillin resistant gene is a gene. Okay. And then like this tetracycline resistant gene is a gene. Ampicillin resistant gene. Tetracycline resistant gene. Like this, many genes are there. Into this gene, we are introducing the foreign gene. If I am introducing the foreign gene into the tetracycline resistant gene, this gene will not make the enzyme to break tetracycline. This gene will not break the enzyme to break tetracycline. Why? Because we already corrupted this gene by introducing the foreign gene of your interest. For example, might be a growth hormone gene, might be an insulin gene, etc. So that foreign gene I introduced into this tetracycline resistant gene. So this tetracycline resistant gene can't make the enzyme which can't make the tetracycline. So this is a way you need to remember the concept in your mind, right? So now we are entering into the next important concept after the cloning side. So you know very well vectors for cloning genes in plants and animals. So this is a classics information for you all. In this particular area, sometime if I need to introduce a foreign gene into the animal cell, I'm using retrovirus. I'm using retrovirus. We know very well retrovirus infects major important site. I'm not using HIV. I'm not using HIV. Okay, your Gotouniversity team told retrovirus HIV is used as a vector. I'm not told. Okay, here the retrovirus, some are having this feature. Okay, so what we are doing is basically we are using influenza virus is totally different from Haemophilus influenza bacteria. Okay, Hari is saying Haemophilus influenza virus. No, Haemophilus influenza is a bacteria. Influenza is a virus. Both are different. Haemophilus influenza bacteria, I'm not discussing in this contest. I'm introducing the influenza virus concept, which is having the foreign gene, which is incorporated into the target DNA, which is incorporated into the target DNA. So into the target DNA, which is incorporated, okay, which is incorporated. So the virus gene is incorporated into the bacterial DNA, the bacterial DNA. Again, back I'm repeating, into the bacteria DNA, we are introducing the foreign gene of our interest uh, by using influenza virus. No, this is a bacterial DNA. This is a bacterial DNA. For example, I need to introduce into the lung cell. Lung cell DNA. and that too it is a bacterial DNA into which I introduce my gene of interest with the help of the influenza virus, which is a retrovirus. All viruses are not retrovirus. All viruses are not retrovirus. Only certain virus or retrovirus having RNA as a genetic material, right? So what we need to understand basically, by introducing the foreign gene of our interest into the target lung cell by using influenza virus DNA, I can make the lung cells to produce yeah, valuable components in such a way the proper gases exchange happens in the lungs to facilitate our gaseous exchange mechanism. Our gaseous exchange mechanism is quite amazing. Why our gases exchange mechanism is quite amazing? Lung cells are doing the work more appropriately. Why? Because the lung cells are having the high quality gene, which is introduced by influenza virus DNA. Influenza virus DNA. So this is a classic example, the way how we are introducing the gene into the animal cell. And the same way, for example, if you're talking about Agrobacterium tumefacium, it's a bacteria into which I am introducing the gene of my interest, and I'm making this bacteria to introduce a foreign gene into the cotton plant. Okay, into the cotton plant, I'm introducing the gene of my interest, okay, with the help of the Agrobacterium DNA. Agrobacterium DNA. So agrobacterium DNA is used to introduce the foreign gene of our interest into the cotton DNA. Into the cotton plant cell DNA, into the cotton plant cell DNA. Again, back I'm repeating, into the cotton plant cell DNA, into the cotton plant cell DNA, into the cotton plant cell DNA. I'm introducing gene of my interest by using agrobacterium DNA. Agrobacterium DNA, right? So in one place, I'm using virus. In one place, I'm using bacteria to introduce a foreign gene into the host DNA, right?